Thank you. Perfect, the mic is working. Thanks. 5 minutes ago it looked like no one's coming. Now room is full. Thanks for dropping by and for interest in that talk. Thank you guys for organizing this. Very, very honored that I got a chance to talk to you in this beautiful setting. And yeah, so I'm Volker, and I am currently based in Munich with Fabian Theis and Alex Wolf. We've been working on this for quite a bit. So basically considering myself now as a computational biologist, even though I'm neither comp nor bio, but my background is actually math and finance. So that's where I'm originally from. And then it was around 2.5 years ago when I made this switch. Was initially kind of challenging, but now I'm actually really grateful to be part of this. And I'm currently here in Boston to work on some further extensions with Peter Kashenko at Harvard Medical, which is I think going really well. And yeah, I'm looking forward. Like I was told, there's a lot of questions, so I'm looking forward to those and also the discussions afterwards. I'll be around afterwards as well. So this is going to be in the context of single cell, obviously. So just super briefly talk about that, and then we dive right into the topic. Single cell, like very popular. Tracing it back to 2013 became the method of the year, right? And since then, like it's now 6 years later, it's quite astonishing how this has been progressing and evolved, and like where we've had it right now. So not just academia, but also in industry, how everything's like flourishing. As a finance guy, I just looked up those numbers, the global market value of single cell analysis. So we're already talking about the billions now, right? So coming from nothing, that's quite a good return in that sense. And especially now as it's paving its way into precision medicine as well, it feels like this is not the end, but just the beginning of like a bigger story. And yeah, so let's just dive right into this. Just that like I get everybody, and I know everybody, so just a very brief intro into single cell. You get a bunch of cells, and then you do sequencing, say droplet based, and then your output. Like, of course, you have like the quality assessments and alignment, everything, but eventually you get a cell by gene matrix, right? So for each particular cell, you get its transcriptome profile with all the instruction of that cell. And there's usually a lot of data, so 30,000 genes across like a large number of cells. And you got to make sense of that. So I have a big data cloud, and usually what we do, we basically reduce those 30,000 gene dimensions into a lower dimension embedding, and we come up with something that's kind of retaining the local structure, maybe also the global structure of the data. So basically having two, like each point representing one cell, 2 points being close by means we've got some like 2 cells that are transcriptomically similar, right? And most of you probably know that our lab has set up this Python toolkit, ScamPy. So I just want to make a short advertisement for that as well. Yeah, we've put a lot of effort into making this scalable and efficient and very user-friendly, so it's kind of nice to play around with that. Hosts a lot of tools, and it's also going to be, like, we're about to extend this towards like multi-model data, also spatial is coming. So it's going to be like up to date with the recent technologies as well. And obviously, like this process of coming to a way where you can kind of like interpret your data, that's obviously very, very simplified. There's like all kinds of steps that you have to look into and be aware of, going from normalization, batch correction, etc., like all the pre-processing steps until you eventually reach some, yeah, like where you can do some downstream analysis. And each step within these like bears its own story and has a lot of issues. And Malte from our lab, he dived into all of this and wrote down this very nice tutorial. So even if you consider yourself an expert in single cell, I think it's really worth looking into this because there's like all kinds of pitfalls. And so for instance, I thought normalization is basically trivial, but it appears to be not so trivial anymore, as there's just been this conference, NormGem in New York, whole conference just hosting for talking about normalization. And then There's obviously other interesting parts, such as batch correction, which feels like still kind of unsolved, and there's still a lot of potential within each of those. So now we want to make sense of this as in kind of do some dynamic inference, and usually how we like approach this is by those concepts of pseudo-temporal ordering. So basically, you kind of view this as a graph of cells being like similar or not so similar to each other, and then you basically route your process. Say you have some stem cells up here, and then you kind of track like by a Markov diffusion walk along the graph, and you kind of generate these trajectories, right? So this is just ordering along their transcriptomic similarity. So it's a bit like here, Götzschen showing off in his holidays. So you kind of make sense of this, this like gravitational law, and you can learn something about it, even though you don't have the video of this, but just snapshots being ordered together, and then you can actually learn something of this, right? But once it comes to something, say, more complex, or if you want to do some lineage decision, if you want to learn something about sulfate commitment, you got to have some additional information, right? So just similarity-based, that won't be sufficient, especially like kind of getting an idea of like what's happening at this branching point, which cells may be already determined to a particular fate. So it would be, of course, nice to get access to this full phase space. So that would be just like basically imposing this error on top of that, and that's the concept of RNA velocity, right? And I think this is really beautiful because there's like very simple math, and the impact is just dramatic. In fact, there's like more than 70 methods already around on dynamic inference, on trajectory inference, and I felt like this may be enough, but then Peter and his colleagues came and with this concept of RNA velocity, and it's just transforming the way how we think about it. All right, so let's consider like just exemplary. This is hippocampal dentate gyrus, and so assume we have done our pre-processing. By the way, like all the results I show, that's like very basic pre-processing. So we do gene filtering and then normalization. That's basically it. And then so here you can basically route your process, start from those NIPCs, and you kind of make sense that they're maturing into those granule cells, right? But what about the other parts? It's kind of hard to get this just from similarity-based methods. So with RNA velocity, you can basically impose from the splicing kinetics, you can impose this directionality on your data, and you can even zoom in and like look at single cell expression and for single cell velocities, and then within these GABA cells, you wouldn't be able to get this just by similarity-based methods, but now you have a clear like sub trajectory in there that you delineate. Okay, briefly about the concept, and then I'll show you how we extended this model. So starting from DNA, it's transcribed with some transcription rate to premature mRNA. So you have introns and exons, so coding, non-coding parts, and then the introns would be spliced out, and you have your exons joined together, right? This is the process of splicing, and then you get mature mRNA, and then you also have eventually degradation, right? Now, what these guys found is that you can, from the standard protocols, you can basically distinguish those two just by looking at the presence of introns. So you can distinguish between unspliced and spliced, just looking whether there's some exon-intron boundaries or where you have introns present. And then you basically have not just this one gene expression, this one modality, but you have two modalities now of spliced and unspliced. And that's basically old and new mRNA. And that gives you an idea of like some kind of temporal sequence within your data. You can very easily formalize this, so as in very like a linear, easy ODE. of unspliced being produced at the transcription rate beta and being spliced with beta times u, and then splicing is now the production of the spliced, and then you have degradation of the gamma S. So the tricky part here is, and that's one of the fundamental differences of what I've been doing back in finance. In finance, we had time series data, so basically you could just track your stock market prices, and you have it like every 10 seconds you have your price, and you can model this very nicely. Here, time is not measured, right? Time is a latent variable, and we would have to infer that as well. And that's the fundamental problem. Where, like, also attributes where why you have so many tools around doing trajectory inference. The reason is very simple, because when you take a measurement is destructive to the cells. So you can only take a measurement once, right? So you have a snapshot of cells. So to overcome this, so we don't know time. To overcome this and not like make this invariant basically of time, there is a very simple idea. So consider the process of, like, say we have 3 hours of transcription. So this is on. So basically you have induction in unspliced first and then spliced as a reaction up until the point where transcription terminates, and then you go into repression phase. And then you have this exponential decay, again unspliced first and then spliced follows, right? So now, under the assumption that you have steady state sampled in here, this can be very, very simplified, and you can basically compute your velocities within 2 equations. Let's see how this works. So having induction, so this corresponds to this part here. So the induction phase up until you reach a steady state. And then you go into the repression phase, right? So you have this bulby, this curvature, because unspliced goes up first and then spliced. So if you know that there is a steady state sampled up here, so you can basically fit a linear regression on those steady states. You actually have 2 of them, so one up here, one down here, right? If transcription takes long enough. So you can basically fit on those extreme quantiles. So this gives you a steady state ratio. So where the ratio of unspliced to spliced is in steady state, where there's a constant transcriptional state. And whatever deviates, that is dynamic, right? So RNA velocity is basically just the residual to the steady state ratio. So the deviance of an observation to the steady state ratio. So this is just to formalize this. We fit a linear regression on this. This is under the assumption that we have a splicing rate shared across genes, because this comes from this equation. So this is RNA velocity, right? The time derivative of S. Since this is not identifiable, we would set beta to 1. So we get a common splicing rate across genes. That's the other assumption that we make here. And then you can basically just compute the velocities as a deviation from the steady state. Okay. Now, everybody can follow me yet? So far, everything's fine. So what is the issue? If we not have 3 hours of transcription, but only 1 hour, it could happen that we don't reach steady state. So there's like an early switch in this repression phase. And what happens is our phase portrait looks more like this. When we look at unspliced versus spliced, the turnover to the repression phase is at an earlier state than, and it has not reached yet steady state. So if we now just fit on the extreme quantiles, we can be quite misleading with the fitted ratio. Another problem, and I think that's also worth mentioning, when we only measuring like parts of the data, so not just this full splicing dynamics, and that happens quite often. When we only measure parts of this, it will also eventually fail to capture this. So there's a couple assumptions that we make, and that could be problematic when we look in particular at systems where there's like rapid changes, some development or response to perturbations. So the drawback is that you would have to have these steady states sampled. We're not actually using all the samples, so it would actually be nice to kind of learn this curvature as well, so that we haven't done here. And yeah, the problem is not identifiable. That means we assume a common splicing rate across genes. Okay, now what can we do? Initially, we started with some like kind of trying to incorporate stochasticity and like all kinds of extensions. And eventually, we thought maybe we could just solve it completely, like including time, including transcriptional states, basically everything, all the parameters in here. So first of all, this ODE is linear ODE is very simple. So you can just solve it, and you get this, right? So there's some terms that describe the off dynamics, some terms that describe the on dynamics, and some that describe the like covariation between splice and unsplice. So there's a lot of parameters, and it feels like it's kind of impossible to get this. So first, we have direction rates. So that's what we're actually interested in, alpha, beta, and gamma. We also account for basal transcription. So the repression phase could not be like completely off, but it could have a basal transcription. And then we have all those latent variables that we're not particularly interested in, but we need those to basically describe the system explicitly, right? And that would be time, so a cell-specific time point. For each cell, we would need to know what transcriptional state does it belong to. And then here we model 4 states, so induction, repression, and the 2 steady states. And then we would need to know at which time point does it change? Where is this transcriptional switch going on? So the idea we had is if you had like all these, so let's say you have time series given, and you know where the switch is happening, and you know like so basically you have the latent variables given, right? Then you could basically infer the other parameters. And vice versa. So if you have the direction rates given, you could actually infer time points. That was a bit like troublesome to get there to make this work, but yeah, there's some tricks. So what you can eventually do is, so it's like the chicken and egg problem. So basically, you solve this with expectation maximization, right? So basically, fixing one set of parameters, solving for the others, fixing the other set of parameters, solving for these, and then iterate until this hopefully converges. It doesn't always converge, just to spoil already. But in the cases that we're interested anyways, there it does. So just briefly, how does it work? First of all, you initialize your system. It's actually initialized with the steady state parameters. And like obviously in the beginning, you get something that doesn't look so nice, that doesn't capture the data so nice. It's kind of a bit off. But basically, you can now project your observation onto this curve, and that gives you a time point because it's explicitly given by the ODE, right? So it's just finding the time that corresponds to this like sample on the phase trajectory, right? So you have time, and then the transcriptional state. We get this by likelihood. So this is all likelihood based. So for each sample, you can basically find the shortest distance to the on curve, to the on state, to the steady states, and also to the off state, and then you get 4 likelihoods, and then you can just optimize across them. So this is actually the E steps you would have to either integrate over those likelihoods, or you just take the map, right? And then you do your parameter update. So there's just basic gradient descent. It's like a momentum-based version to make it a bit more like faster to converge. But it's just gradient descent, so we basically optimize our loss function, which stems from the likelihood. So the likelihood, we can just easily write it down. And if you assume a constant variance across all samples, so this is all for one gene, right? If you assume across all your samples the same variance, of the residuals, then this basically reduces to a mean squared error. So it's actually very simple to optimize.
Question? Are the projections all when you do the time assignment?
Oh, that's a good point, yeah.
Does it still hold the total transcripts constant, or what does that happen at the 45-degree angle?
So actually, the projection we would have an orthogonal projection, right? That would be the optimal optimal time assignment, like the shortest distance to the curve. So what we did here is this is just to make things more efficient. Basically, you can write down S as a function of U, so splice as a function of U and time, and then you solve for time. You basically take the inverse of a linear combination of S and U. So there's like an explicit way of phrasing time as a function of S and U, and then you get an explicit way of computing time. That's obviously very, like, much more efficient than solving the whole optimization problem. So that's what we do up to the very last iteration. In the very last iteration, we actually solve this then likelihood-based. It's just for efficiency reason, and yeah, it's good that you point this out. Yeah.
And is it a set of parameters for every gene, or do you have?
This is all per gene. Yeah. So it is completely decoupled yet. Yeah.
So is there any sharing along genes? Because
I'll tell you later. Yeah. Yeah. Not so much yet, but we have like, yeah. I'll tell you.
What's the noise model here? Because you say you have a full likelihood, so it's like Gaussian noise on those?
Yeah, it's Gaussian noise. Yeah. We validated it, and it fits. Like, even on real data, it looks fine. Yeah. Yeah. We also have tried other noise models, but it doesn't really make a difference.
So if I'm trying to project the center of a circle to the circle, I have many choices possible.
Yeah.
So how do you resolve the uncertainties when you project onto a curve from the inside? Like, there could be an entire segment that's the closest point to this point. Just let epsilon machine decide for you.
So in this case, there's not really ambiguity, right? Because it's very clear that this belongs to the on phase. But let's say if you have only this part captured, this part sampled, like, as long as you have a bit of curvature, it learns that, and it signs it properly. But when you have like
the moment where you decide what time you're on, right? So you have to project all those points onto the curve, right?
Exactly.
To decide which time it is.
Yeah.
But if your curve looks like a circle and you're at the center of the circle, there's many choices. Like, there's an infinite number of choices where you could project it.
if your curve, okay, but your curve will never look like a circle. It will always have this kind of shape because it's the way.
I'm giving you an extreme example.
Yeah.
There's many points that project at least 2 points on this curve, right?
Yeah. So if you have, let's say, a point exactly in the center, yeah, it could be ambiguous. So it's not. Yeah, and so this is like every point, like every sample gets a likelihood. So it will get a lower likelihood, especially as we normalize across like the state likelihoods, and then it like intrinsically accounts for uncertainty as well. Yeah. I'll show you a couple examples on real data later where this happens actually. Yeah.
How do you account for a gene that could go in the population sort of on and off and on and off?
Yeah, we don't. Yeah, it doesn't account for that. So this is the assumption. We have one splicing parameter for all the samples per gene, one degradation rate, and just those two alphas. So if we want to do multi-state, I think from the data, like just from unspliced spliced modalities, this is kind of challenging. So you would actually need some time series data if you want to do that.
You need to integrate across more genes, probably.
Yeah, and share across genes, exactly. Yeah, but we don't do that here. That would be a nice follow-up.
To follow up on that question, like if you have like different population in your snapshot, do you use only the cells that are close to your so for each cell to estimate this, you're using the close by cells. If you use all layers like two, let's say the genes doing different things, you will have different models. You know what I mean?
Can you phrase it?
Yeah. So what I'm saying, like for each cell,
yeah,
like are you using just the information of the single cell, or are you using like a local neighborhood functions, like to infer your velocity? Because in Romano they do like
it's smoothed. Yeah, right. It's smoothed across between 50 and 30 neighbors. Yeah. Else you wouldn't get like. You could run this model on raw data as well. It works not as nice, but it works. But you don't visually you just don't see anything. Like you don't get sense of what's happening there. But it's smooth, but it's not as smooth as like I don't know. It's not like 500 neighbors. It's a rather small neighborhood.
Yeah, because I think it's a critical point. Is this parameter totally change as you change this neighborhood, right?
Yeah.
I was when maybe you will touch on this later, but this you will, how stable is your model that you change.
I won't have that here, but we can discuss it later. Yeah.
Are you using like the mean error or like the final likelihood to report some kind of uncertainty or goodness of fit in the end?
Yeah, and we can actually use that for some very nice purposes. I'll also show that. Yeah. Okay, so I'll continue with. So I want to make the statement that we can now basically, so the velocities that we get, they're insensitive to the switching. So we can actually learn the switching. That means we're not reliant on any steady states, right? So we can also observe fit basically unobserved steady states. So in that case, the steady state would lie somewhere here, and we actually find it just by learning this curvature, right? So just simulating, I think it's 2,000 genes, log normally distributed parameters, and then time from a Poisson law. And then if we now look, if we vary the time of transcriptional induction, so between 2 and 10 hours, 10 hours would mean we would very likely reach steady state. In 2 hours, probably not. You see that the error in the estimate can systematically go up with the steady state model, while the dynamical model is completely insensitive, right? And in terms of correlation of the estimated ratio between the true ratio, we get a bit of an improvement in here. Now, the interesting part is that we have just estimated these kinetic parameters, right? And that would be really nice to see whether that actually works out. So basically fitting, visualizing the true parameter, because this is also, again, on simulated data. We're still looking for some real data where we can validate this. So the true parameter against the estimated one, and we get a fairly good correlation. And we get access to 2 parameters, identifiable, say degradation and transcription rate. And the third one we get if we now constrain that, because time is now so far time is learned gene specific. For each gene we have a time course. But in fact time is stems, so this is basically underlying universal time, right? So we can basically constrain that all the time courses for the genes, they should be the same. So they should, first of all, have the same length. So the overall time course should be the same. Then, by unifying this to a gene share time, we identify even the splicing rate, so even the third rate, up to a global scale parameter. So there's one scale parameter across all the genes and all the parameters, some lambda. And if we now additionally impose that we knew how long our time course would take, say 20 hours, then we even identify this in absolute values. I think it's kind of cool that we get access to this. And now we can use this, because now we have an explicit description of the system of both unspliced and spliced, and basically RNA velocity we just get by plugging in, right? Now see how this looks like on the data set that I've shown you before. So first of all, this global lineage of NIPCs differentiating to those granule cells. And then also the lineage up here, radioglide to astrocytes, you get this with both the models, both steady state as well as the dynamical model. So where you see fundamental differences, though, is when you now look at particular cell types that are kind of like far away from the actual lineage. Because in the steady state model, you basically learn, you fit an extreme quanta and then take the residuals. And these could actually pop up as outliers. Right? And that's where this stems from, this noisy velocities. So so basically whenever you want to figure out where your projected velocities come from, just look into those phase portraits, because that's where they stem from. And here you see how you have this upregulation up to, so this basically describing this lineage to the neuroblast, and then going into repression phase, so basically reaching the granule mature cells here, right? Now, if you look at another gene, this FAM155A, then again, fitting on the extreme quantile gives you now the residuals, but the residuals of the Cajalaricio cell, these ones here, they pop out as outliers, right? So if you now apply the dynamical model, okay, you can't see it because it's not there. It doesn't have any magnitude in here. Basically, it assigns like a very, very low likelihood to those because they cannot be explained by the transient state down here, right? So they get a very low likelihood, and they're going to be assigned to a steady state in that case. That's why you have the dynamical model identifies these as terminal, while with the steady state you get kind of noisy errors. In terms of consistency, so this is just like some kind of self-check, self-consistency score, basically saying how does a velocity correlate with the neighborhood, with the neighboring velocities, and you get a score which is like fairly higher because it's just likelihood-based and it's not so prone to noise. Now, coming to your question, what's interesting is since we have a likelihood-based model, we can use those likelihoods to find genes that have these nice display these nice kinetics, right? So apart from like by basically finding this ball rolling down, we basically want to identify those drivers. So we can aggregate across cells to gene likelihoods. So for every gene, we get like an overall likelihood. And now, if we just pick the top ones, we get something like this. So something where it actually displays this nice kinetics in these genes, some in up and down regulation, some just down regulation. And actually we get a very nice convergence. So it's a global convergence with likelihood up to 0.7. While if you just randomly pick any genes, or like these genes are somewhere from the lower quantile of the likelihoods, you just find pure noise, right? So those genes will not really contribute to the velocities. And for those, it might happen that you don't get convergence because there's a lot of uncertainty, ambiguity in there, right? And the likelihood will be fairly low. So in that case, the optimum is actually at 0.1. And I think that's really nice because this is kind of an alternative to this differential expression paradigm. Just that you add this temporal resolution in here. What's also very nice about this, obviously we wanted to validate whether these genes are actually driving this. I think experimentally, I'm not sure how we can do that. We're thinking about it. But at least computationally, what we did is, so let's say you just take the top 30 genes and you try to reconstruct the velocity, the vector field that comes from the velocities. And it turns out that you can actually perfectly reconstruct it just with the top 30 genes, right? If you take all the other genes, except the top 30s, like all the other, say, 8,000 genes, it totally breaks down. So it turns out that those genes actually drive the dynamics, at least in terms of splicing dynamics. Yeah, sure.
So you're measuring all of this based on 3' UTR tag transcription, right? So
yep.
Like your efficiency of capturing introns really depends on how close they are to the 3' UTR.
Yes.
So basically, maybe you can say something about some genes where the introns are well placed, but certainly you can't say if the 3' UTR or the first intron is very far away, you're very unlikely to observe it.
Yeah, sure. It's definitely biased to that. Yeah. So that's
So any claim about these being drivers, right, needs to be heavily caveated by these are the drivers I can measure.
Yeah, that's why that's called a putative drivers. Good. Yeah. Thanks. Yeah, but you're right. Yeah. Yeah, I have to be careful. Now let's have a look at another data set. So you have differentiated. So this is pancreas endocrinogenesis. And you have 4 different fates. So it's the glucose-producing alpha cells. Then you have beta cells that are producing insulin. And then you also have these delta and epsilon cells, right? And we know that the lineage is going like somewhat like this towards those different fates. So there's a very well studied data sets where we actually know like all the kind of biology, which is kind of interesting because I'll show you a few results on that. So now as you apply the steady state model, it kind of captures this overall dynamic, but you get like a bit of backflow here. It's kind of unclear what's happening here. So you get the global dynamics, but you get a bit of like incongruous flow. And if you want to like try to understand where this comes from, just again look at your driver, like the genes that is driving the velocity, like that explains most of the velocities in the embedding. Phrase it this way. And then you find actually, so as you fit on the extreme quanta, you basically cut the alpha cells here, and you cut them so that they're basically lying in induction and repression phase, even though this looks more like this is all induction, right? And reaching some steady state maybe here. So basically these come with a negative residual and cause this backflow here. Now with the I have to say that this is not like very, very smooth. So in the original model, you would like take a lot more neighbors, and then this may be smoothed out, right? But this is just to do a fair comparison again for 30 neighbors. Now with the dynamical model, you get a fairly smooth vector field. And this is without like any additional smoothing. So this comes purely from the single cell velocities. And if you again look at the gene-wise velocities, you find that these will be mostly contributing to being assigned to a steady state, right? Now here, first I thought this is a bug, but after validating both experimentally and then also looking at cell cycle scores, it turned out this is actually a cell cycle. So it's kind of nice that it delineates this, and we kind of get this exit, and then the differentiation towards eventually cell fate commitment. Okay, so far I have only used kinetic parameters and basically the description of our system for these velocities, but there's a lot more that we've digged out, right? Because there's this time, and there's also transcriptional states, and there's this switching time. So I was wondering whether we can also use those to kind of get an idea of like get some information from the data in here. And so what you can do is, and this is again this unified gene share time. So how do we do this? It's basically some kind of median of time increments. So all the time increments, you just take some sort of median across all the genes, and then what happens implicitly is that so for one gene you have this late turn on for that particular cell here. In this downstream, you also have a late time point. But here it's not so clear, so it's kind of ambiguous whether this is going to be early or late. But by imposing from the other genes, you basically make a decision for late, right? So this is how you couple the gene-specific time points to a one universal time point that is rooted in a particular cell that is inferred from the vector field itself. So you don't need like a user-specified root cell, but it identifies itself. And then we can use this. So this is just some sanity check on simulated data again, where you have this kind of like very, very clear that dynamics. And if you apply pseudo-time, because of similarity base, you won't capture this in here, right? So this will fail. Now, as you apply this latent time that's purely based on transcriptional dynamics, so it's it's completely independent of the similarity, connectivities or anything. So it captures this almost perfectly as a perfect correlation. Okay, so now can we use that on real data? Let's have a look. So the thing is, on this data, we know basically a lot of stuff. So we know that alpha cells are being produced at a time point of like embryonic day 9 to 12. And then from embryonic day 12 to around 15 and thereafter, we have beta cells being produced. So there's like a clear sequential ordering sequence of those fates, right? So if you now apply pseudotime, so just rooting it somewhere up here, and then basically walking along this graph, and then this gives you a distance, right, from the root cell along the graph, and then, but beta and alpha cells, they're not really distinguished in here, so they're treated the same way. Apart from that, it looks fine. Like this looks like it can delineate it very well. Now, what happens if you apply latent time on this now? And again, this is just really simple. Like after doing all the fitting, it's just some kind of big normalization effort, and then you get this. So it's actually also very efficient. So what latent time does, it actually relates those fates, right? And you actually figure out that this comes first, and then the beta cells rise up later. Can you see that? Maybe a bit. Okay, now another parameter that was interesting, because we learned that the time course, but we also have these switching time points, basically where there's a transcriptionist switch going from induction to repression, or say from induction to homeostasis. And so what we can do is just count the number of switches across genes, right? So how many genes are turning at a particular point, and then we can just impose this coloring on top of the embedding. And what we find is actually exactly like where this self-fate commitment happens, where you have some branching point, another branching point. Exactly at those regions, you have a lot of genes that are confidently transcriptionally switching, right? Now again, you can use gene likelihoods, and so now we not only take the top gene likelihoods across the whole data, but you can actually constrain it to a particular region in your data that you're interested in. So I want to know what are the genes that show, like, that displays very nice kinetics, for instance, down here at the branching point. So you can basically compute the gene likelihood just here. And yeah, and this is just visualize these genes, these dynamical genes along the latent time, where you see those cascades. And now for each of these regions, taking the top likelihood gene, we find these genes. So each gene displaying some kind of kinetics within one of those regions. And we can, again, display this along latent time to see where the switch is going and how fast and so on, and get an idea of where this lineage commitment is happening. And what are the genes that are turning in the branching point. Okay, this is so far, yeah, I think I'll stop here for a second. Is there any questions? Yeah.
Yeah, I'm really a big fan of the XBRL.
Thank you.
When we're using it for our own data, when we're building the latent time, pseudo time, sometimes we can find some inconsistency. For example, for the root cell we identify.
Yeah.
For example, we identify the turnout state at the starting point and the left, we can observe the latent time. The starting point would be the right end of the cell.
Yeah, exactly.
So how could we solve this problem?
So the way how we compute the roots right now is very, very simple. So we haven't done like we haven't put a lot of thoughts into this yet. I think it can be done better. So it's basically you take the transition matrix and compute the left eigenvector, and that gives you a terminal state, right? It's really simple. And for this, you can, like, it's basically a one-liner. And then you can, yeah, but then you rely on these root cells to be the right one. So what happens with the current implementation is that sometimes you get root cells like some are basically isolated cells as also being identified as root cells. And that's really an issue right now. So you have to check that you get the right root cells, else you won't get the correct latent time. So because the problem with isolated cells is there's no outgoing flow, and then it identifies those as root cells as well. So I think it shouldn't be too hard to make this more robust, but we're working on that.
Oh, thank you.
Yeah. Yeah. And actually, we already implemented a version that not only like looks at those time increments starting from the root cells, but also from the endpoints, because endpoints are a bit like more robust. They can be more robustly inferred for some reason. Yeah. And then this becomes a bit more robust. Yeah.
Endpoint modeling would be more robust than the root cell modeling in terms of the latent time.
Yeah, exactly. Yeah.
I have another question. Sometimes you see this connected population, right? How many cells do you need to reliably estimate the velocity? In some area you have very few cells. Can we trust this area? Like again, how many cells do we need, to estimate this velocity?
So most of the issues arise exactly when we have only very few cells, because there's a lot of ambiguity yeah, say you have just kind of a straight line that could be both. It could be assigned to induction, but it could equally be assigned to repression. And we're currently working on some ways to basically use some additional information and also do it shared across genes to get this more, like also for small sample size. But that's an issue. Yeah.
Do you have a rule of thumb, like you need at least 10 cells, or 50 cells, or how many cells?
So 10 cells will definitely be not enough. Yeah, there's no rule of thumb, but you got to look into the phase portraits and make sense of what it has inferred, and then yeah, evaluate from there.
Yeah, and another comment, because we're trying this tool and we're really excited.
Yeah.
So we did a simple experiment, taking the same sample and resampling, right? Splitting in two and then just comparing. Sometimes you see that arrows totally change direction, even in the area where you have like a lot of cells. So I was wondering if you have any suggestion.
So they change because of what?
So essentially my ground truth is I take one sample and then I resample the same sample, just split, let's say, in two, assuming that you have enough cells, right? And in areas where you have enough cells, you see that some of the arrows change direction.
Yeah.
So and then some really consistent, right? But some totally change direction. So I was wondering if you had thought about some metric to say, okay, you can trust this area well based on resampling or other strategies.
Yeah, we have that implemented. So there's all kinds of confidence measures. And so the issue that you were now referring could be due to some batch effects, or could be due to some, I don't know, alternative splicing, because one population is just bearing some totally different splicing kinetics, which is not connected to the other one as well. Yeah, it could be a lot of reasons. And then again, you would have to look into the phase portraits. And I think it's important to kind of, especially if you know, if you have your batch labels, to do this per batch and analyze this in a very, yeah.
Also, we would like to know the saturation point. For example, how many of the cell number or how many of the gene number with high likelihood would obtain a very consistent trajectory. Yeah. What would be your suggestion?
What's the number of?
What the number of the gene, what the number of the cell? For example, we have 5,000 number of cells, then we will confidently claim this is the successful trajectory, or successful transient dynamic modeling.
Yeah.
So what would be your suggestion?
So for the number of cells, just take as many as you can, obviously. But it's important to split those into the different batches. So if you're very sure that you have some batch effects in there, then it's important to treat them differently, to treat them separately. And for the number of genes, it shouldn't matter that much, because as long you kind of capture the top genes that are driving this, it's fine. And like the usual filtering we do is like the top 2,000 highly variable genes, and that is totally fine. Yeah. So I just continue with a couple more slides, and then I take more questions. We have thought about also accounting for stochasticity, and in particular when we have small sample sizes, this can be very, very interesting because it also contains some valuable information. So that basically is, I won't go too much into detail, just give you a brief idea. That is basically just treating these kinetic events of transcription, splicing, degradation as probabilistic events. And then what you get, you can basically write down your chemical master equation. You can write down all the moments, basically all of them. And as you, like if you just look at the first and second order moment, you get a moment closure. You get a closed system, and like everything is linearly dependent and very nice. And so this is what we've already seen. So this is what we know. And this is basically the what you get from second order moments. So you also make use of variances and covariation between unspliced and spliced. And to incorporate that in a dynamical model is not so straightforward, and we haven't done it yet, but we're on it. But basically using it to estimate steady states, so basically extending the steady state model, that is fairly easy because it's also just a linear regression on not only on the first order moments, but also on those covariance terms. And interestingly, so I thought this won't give us much because, like, yeah, it's not so sure like whether this helps at all. But eventually we just computed these consistencies again. So basically the correlation of one velocity with their neighboring velocities. And it turns out it actually improves self-consistency. So the vector field becomes more coherent. And there's like a couple of genes where you can actually see that it better captures these steady states. All right, cool. So that's interesting. And now we have implemented all of these models, both like the steady state, also the stochastic, as well as the dynamical model, and put a lot of effort into making this efficient, also with the help from the community. This has really shaped this. It's actually been released a bit earlier than our actual preprint, which was not planned, but turned out to be kind of helpful because we got a lot of feedback, a lot of input, and we're able to make this not only scalable, but also efficient. So basically, when you run 30,000 cells, you can do that in less than a minute, like running the whole pipeline, starting from your pre-processing all the way to projecting the velocities for the steady state model. The full dynamics is obviously a bit more expensive, so that would be around 30 minutes, but you just let it run for 30 minutes and you get like all kinds of information. And you also know what are the genes that are contributing to these projected velocities. I think that's kind of nice. And that is due to some the integration with ScamPys, some parallelized pipelines. We use some efficient nearest neighbor search, analytical closed form solutions. So whenever it works out that we can do something analytically, we tried doing that, so that accounts, for instance, for this time assignment. Also the switching time we could get analytically, and then yeah, steady state and stochastic model anyways. And I thought this is kind of nice to show. This is like this these guys from where is it? In Lund in Sweden. So it's just two weeks after our preprint was out, so they put it into VR, and I think it's kind of fancy to also see this velocity stream in 3 dimensions. Actually on 140,000 cells, I think that's a cool number. Cool to see that it actually works out on a larger cell number. Yeah, I have already mentioned a couple of these, just the potential limitations. So alternative splicing is an issue. Indeed, so what you usually see is like 2 different slopes, and then you can be sure that this is either attributed to alternative splicing or there are some modulations like in degradation. that can be quite an issue. Like, because it's basically causing in the end some differential gene regulation, right? And then, like in the worst case, it can even happen that in the phase portrait, the directionality just exactly turns over. When, for instance, I've just discussed this yesterday with Ruslan. It could actually happen that directionality exactly goes into the opposite when, for instance, the gradation is slowly going up. It's like a time-dependent variables that is increasing along the trajectory. So it's kind of hard to control for this, but we've put like some thresholds so that these would be filtered out. For now, I think that's okay. For the future, it would be nice to address these issues. Then this ambiguity in state assignment. So here, just looking at one gene, it's not so clear whether this is like really some downstream flow or whether this is actually contribute to some induction phase. So right now, the only way like where we couple genes is through this latent time, but I think it can be done even better than this. And then this is not an issue in general, but particularly when you have very small cell sizes, or let's say it's like very isolated cells that are not connected to the main trajectory. It could happen that your velocity projection is kind of unreliable. So that what does it mean? The way velocities are projected is you have your velocity vector in high-dimensional space, and you just check which of the cell-to-cell transitions correlate with this velocity vector, right? So you get cell-to-cell transition probabilities, and these you can then use to do the expected displacement in the embedding with respect to these transition probabilities, right? So this is how the projection works, and that could fail if, say, if these cells just don't exist, right? If you don't have anything matching the velocity vector. So that can also be an issue, but this is usually only the case for smaller cell numbers. Oh yeah, and for this we have all kinds of confidence measures. So I wanted to make sure that like you don't interpret anything that could be like just wrong. So we put a lot of like additional tools like exploratory analysis in this pipeline that you could try out and see like whether your velocity projection is confident at all, and whether your velocities in high-dimensional space are also confident at all. Yeah, so I want to conclude with having this generalized to transient population where you might not capture any steady states, or where you only have like parts of the splicing dynamics, then I think it's kind of nice to identify those genes that contribute to the velocity projection. It's kind of able to capture cell cycles and all kind of stuff, and then latent time gives you another angle of basically being able to relate those different lineages and cell fates. So for now, we have constant reaction rates. It would, of course, be nice to make them time and state dependent, to incorporate some gene structure that can particularly help when we have like genes where it's very ambiguous what's happening in there. Also account for alternative splicing. I think incorporate metabolic labeling and these parts that's already partly implemented but not yet sold. Yeah, I think we have to do it in a more robust fashion, but we're basically working on this. And then, yeah, there's all kinds of other follow-ups and extensions that you can do. Just to mention one more as an outlook, that's what we're currently working on. Since our lab is very exposed to deep learning, to do some kind of neural net architectures, it would be nice to kind of bridge these two worlds. So we have this interpretable mechanistic model, but it's also very powerful to have those autoencoder type of models. And so this is just an excerpt of this by Mohammad and Alex from our lab, using an autoencoder combined with vector arithmetics and latent space. So you basically go in latent space and you find, you build it in such a way that you can basically estimate these perturbation effects. Basically get this delta vector that explains the shift between two conditions, say control and simulated. And you basically train it on some of them, and then you can like go back to a high-dimensional space and predict what would be the counterpart, the stimulated counterpart of this cell type here. And often we see that there's like this one cluster, and then there's another cell type, and we know that it stems actually from this one, but we miss the bridge, right? So we don't know how it went there. And it would be nice to do this kind of out of sampling, generate some trajectories on not observed, on unobserved data. Oh yeah, with that I want to thank you. Thanks. My lab is a lot of fun working with these guys. If you happen to be in Munich, come by and visit us. I want to particularly thank Fabian and Alex. So these been mainly working on that. It's really been a team effort. Also Maris and Stefan, who's been contributing to this. I got a lot of biological feedback from Malte and Sophie and Mohammad. And then also want to thank Peter, where I'm right now working on some further extensions, and also Sten for all kinds of stimulating discussions. And yeah, thank you for your attention.